So we're going to start talking about the cell. Alright, so first we're going to discuss microscopy, and then we're going to move on to talking about different types of cells and some of their structures. Alright, so kind of reiterate a point that was made in chapter 1. Alright, so every single living organism, alright, every single organism that we classify as living is made up of cells. Alright, so this is why the cell is referred to as the fundamental unit of life. Alright, so when you're talking about things like biological molecules, and you're talking about atoms, and you're talking about organelles, alright, all these things are components that make up a cell, alright, but they are not living. Alright, so cells are the fundamental unit of life. Alright, they are the level of classification on the biological hierarchy that we consider living things to first exist. Alright, so one of the major key components of this chapter is kind of getting a handle on what all the different structures, components, and organelles of a cell do. Alright, and which cells have which components. And this is something we'll come back to, but all cells, alright, have certain features that they all have in common. Alright, if you're talking about a eukaryotic, you're talking about a prokaryotic cell, or you're talking about different types of eukaryotic cells, there are certain shared features, alright, but there are many differences between the cells which makes them different from one another. So, like, for instance, talking about a intestinal epithelial cell versus a skin cell versus a skeletal muscle cell or a cardiac muscle cell. Alright, all these cells are very different. They're different in their shape, they're different in the types of organelles they have, they're different in their function, and all this is tied in together. Alright, so come back to this idea of structure and function and what components are found in a higher proportion in a type of cell. It's going to be related to what that cell does, what its ultimate function and job is. Alright, so most cells, alright, so besides, for instance, the largest cell I believe has been characterized as like an ostrich egg. That may have changed recently, but that is the largest cell. Alright, just think about it. When a sperm fuses with an egg, alright, it forms one cell. Alright, before that cell begins dividing, it's one cell. Alright, so think about an egg cell. Alright, think about this egg, this ostrich egg. It's one big, massive cell. Alright, before it starts dividing and forming the little baby ostrich. Alright, but most cells, alright, it requires the use of using some type of microscope. So we'll talk about a couple of different types of microscopy. Your book goes into greater detail into all the different types of microscopes that there are. I'm only going to focus on a few types. Okay, and those are the ones that I'm going to hold you responsible for when it comes to test material. Alright, so the first type of microscope we're going to discuss is the light microscope. Alright, so in this case, you have some type of light source. Alright, so here we're using a light bulb in terms of visible light. And basically this light source is used for illumination of an object. Alright, so basically when you're looking at your microscope, alright, you have, in terms of a light microscope, you have two sets of lenses. Alright, so you have a lens here that we call the ocular lens. All right. Here, all right, you have basically something that we call the objective lens. All right, so on most microscopes, your ocular lenses have a power magnification of 10x, and then your objective lenses are going to vary. All right, so they'll go from basically 4x to 10x to 40x to up to 100x. All right, and that's basically the limit on your light microscope is up to 100x objective. All right, so when you're talking about microscopes, all right, you're talking about magnification. All right, there's something that you have to become familiar with, and this is something called total magnification. All right, so this is basically you take your ocular power and multiply it by your objective power, and that gives you your total magnification. All right, so for instance, if you have your 10x ocular lens multiplied by your 4x objective lens, well, then your total magnification would be 40 times magnification. All right, so whenever you're looking at an object through the ocular lens, and the light rays are also passing through that objective lens, you're magnifying the image 40 times. 40 times what you see with the naked eye. Okay. Now, these things are referred to as now most of these guys are compound light microscopes because you have two sets of lenses. You have objective and ocular lenses. All right. Now, what's happening as your light rays pass through your specimen on your slide? Well, here, all right, you have your little bitty object on your slide. The light rays pass through your object, and as the light rays pass through the objective lenses, all right, your lenses are convex. All right, so what that does is it causes the light rays that pass through the specimen to spread out. All right, and basically what you see with your eyes is actually a magnified virtual image. And in fact, what you'll see is that the object that you're looking at under the microscope and what it looks like on the slide itself is actually going to be backwards. So it's going to be backwards and it's going to be inverted. All right, so what's really left is really right, and what's really up is really down. All right, so when we get to the microscope exercise, this will become much more clear. All right. Now, when you're talking about microscopy, there are three sets of terms you need to become familiar with. All right, so magnification. All right, so you're talking about a ratio of an object's image size to its real size. All right, so basically you're talking about, the relative size of the image under the microscope compared to what it is in real life. All right, so resolution. Resolution has to do with the clarity of an image. All right, so basically the level of detail you have out of it. All right, so the ability to distinguish two separate points as two distinct points. All right, if you have poor resolution and you have 2 points, all right, two individually distinct points. All right, if you have poor resolution, you're not going to see those two individual points. You're going to see like a blurred smudge, all right, that may look like 1 point. And then contrast. All right, so whenever you're looking at a sample, all right, under the microscope, cells themselves have their own level of contrast, all right, because the cell has components inside of it, and so there's going to be areas that are really light, areas that are really dark, depending on how concentrated certain things are in a cell. And obviously, if you're talking about a live cell, these things are going to be in constant motion, so they're going to be ever changing. All right, but we have ways of adding contrast to a specimen on a slide. All right, so here, looking at magnification. All right, so you're looking at the effect of magnification by itself and the effect of magnification with resolution. All right, so here you have a specimen at 10x, specimen at 45x, and specimen at 100x. And notice that as you increase the power of magnification, what you're doing is you're actually getting a closer image of a smaller part of your sample. All right, so that means you have to actually move around the slide to get the whole picture. All right, but what you see under a higher power of magnification is finer and finer details. Now, you notice that as you increase magnification by itself, all right, the image begins to blur. All right, but if you have resolution, all right, the image becomes much more clear. And you can see the finer details. You can see all the cellular components, all the internal structures. All right, so this figure is kind of giving you a representation of different objects that you can see with the naked eye, the light microscope, and with an electron microscope. All right, so we can look at other individuals in a room and note their height, kind of get a gauge and approximation of how tall they are. All right. You can look at chicken eggs, you can look at frog eggs, you can look at human egg. All right, and this can be pretty much the limit of our ability to see things with the naked eye. Okay, then you'll need a light microscope. All right, so we can look at things in finer detail. So we can actually look at what's going on inside of an individual cell using a light microscope. All right, we can look at some of the cellular components. All right, you can look at most bacterial cells using a light microscope. But then if you want to see other types of organelles and other types of smaller cellular structures, you actually have to use something called an electron microscope. All right, so if you want to see really teeny tiny bacteria, especially teeny tiny bacteria that are intracellular bacteria and completely intracellular, if you want to look at viruses, if you want to look at other organelle or cellular structures like ribosomes, or you want to look at proteins or lipids, all right, you need electron microscopes. And then another type of microscope that's not on here is referred to as an atomic force microscope. Okay, so this atomic force microscope will actually allow you to see individual molecules and even allow you to see individual atoms that make up a molecule. Alright, so each type of microscope has its own advantages, disadvantages, and uses. Alright, there's only so far that a microscope will allow you to see certain types of objects or structures or cells or components. Alright, so talking about electron microscopes. So you have two different types. You have scanning and transmission electron microscopy. Alright, so in this case, instead of using visible light as a light source, what you're using to basically look at the specimen is you're using an electron beam. Alright, so you're using electrons, and electrons are actually hitting your specimen. Alright, so scanning electron microscopy is really good at looking at the overall surface of some type of cell. Alright, getting kind of an idea of its shape and what it looks like. transmission electron microscopy is a whole lot better at looking at internal features of a cell. Alright, so here you're looking at a comparison between a light microscope and an electron microscope. Alright, so just like your light microscope, your electron microscope has a series of lenses. Alright, but here what you have is an electron source up top instead of a light source down at the bottom. Alright, your electron source is usually something like a tungsten metal that's basically giving off electrons, and this electron beam is going to be focused on your specimen. And these electrons, what they're going to do is they're going to hit your specimen, and when they hit your specimen has been coated with a type of metallic stain. This metallic stain will allow for these electrons to bounce back off, and it gives some contrast, right? Because they're not going to pass right through the specimen. They're going to bounce off, and they're going to bounce off, and you're going to capture that image, and you're going to get basically a projected image of what you see on a screen, on a computer screen nowadays. Older models of electron microscopes have a photographic plate of what you're looking at. Right, nowadays, most electron microscopes use computers as your display monitor. Alright, so we also have another type of microscope called a phase contrast microscope. Alright, so in this case, the idea with phase contrast is you're basically controlling and regulating light as it passes through your specimen. So the light rays are actually going to pass around the specimen and through it, and you're going to get different levels of contrast, shading, and this allow you to see internal features a little bit more clearly. Alright, so look at a product of these different types of microscopes. So you have a light microscope. So this is a little bitty paramecium. Alright, so one of your little protists. Alright, so the little bitty paramecium, the light microscope. Alright, you can see a specimen. Alright, you can look at a specimen that's living. You can look at a specimen that's dead. You don't necessarily have to stain this guy. You can. If it's a dead specimen, you can stain it. Alright, if it's alive, you don't necessarily have to stain it. Alright, you probably won't. Alright, because you'll probably wind up killing it. Alright, but here you can see the overall shape. You can see some of the internal features. Alright, so you can see these little spherical structures that are motivated vacuoles. Alright, you can see basically this little component here, which is basically a gullet, basically the mouth of the little guy. You can't really see the external appendages, alright, very, very faintly. You'd have to increase the contrast to see these, but you can see little bitty hairs, right? These little hairs, if you look at the SEM image, scanning electron microscope image, you basically see the external features of a cell. You can see these little hair-like projections, alright, so you can see the cilia, which are used for motility, and you can see the gullet, or the mouth, the opening, for which food material comes in, and waste products go out. Alright, your transmission electron microscope. So in this case, you're looking at basically a cross section, and you're looking at all the internal components. Alright, so here in the center, alright, you can see a nucleus. You can see a series of vacuoles, probably food vacuoles. Alright, you can see the membrane of your little paramecium here. Alright, on the outer edge, you can see your little membrane. And very, very faintly, you can see very tiny projections of cilia. And you have other internal features like mitochondria, and should probably be right here. There's a little mitochondria. You can see ribosomes. You can see the ER. Alright, and then looking at a phase contrast. So here, phase contrast, you're usually dealing with live specimens. Alright, but you can see a lot of the internal features of a cell using a phase contrast microscope. Alright, so here, you can see the cilia very, very faintly here on the outer edge. And they're actually moving and they're beating. Alright, so you can see the movement of the cilia. You can see these guys moving around. You can see all the little vesicles, alright, all the little storage vesicles, all the little food vacuoles. You can see the little gullet this guy has. Alright, so there are all these different new techniques, microscopic techniques of looking at cells and looking at tissues in ways in which you can actually label and target a certain cellular structure. Alright, if you want to nowadays, you can label and tag an individual molecule. Alright, and you can basically see where that molecule is transported to in a cell. You can see where it's going. You can map it, and you can actually map it in real time. You can make videos of microscopic images and see where things are moving. You can tag stuff and see where it's transported. Or you can see how individual cellular components are interacting. I mean, it's a totally new world in terms of advances in microscopy nowadays. Alright, so here, talking about tagging structures. Alright, so here we have your fluorescent microscopy. Alright, so confocal fluorescent microscopy. Alright, so basically, you're using visible light, but you're using different spectrums of the visible lights, different wavelengths. Sorry, different wavelengths of the visible light spectrum. Get that a little backwards there. But you're using different wavelengths of the visible light spectrum, and you're focusing these wavelengths on a certain type of component, cellular component that you've tagged with some type of fluorescent chemical. And hit with the right wavelength, that tag will emit back fluorescence and give off color, and that's what you'll see. Alright, so here in the image, alright, you have basically an image of a plant cell here. Alright, so you're looking at the surface of a leaf. Alright, and you can see the little bitty epithelial cells. The epithelial cells are staying in green, alright, on the surface. Alright, and then in red, you have these structures that we call stoma. And these stoma are the openings, the pores, for which water evaporates out of. Alright, so going back to the idea of transpiration, we talked about when we talked about the properties of water, but also this is how gases enter and leave the leaf. Is through these stoma, these openings on the surface. Alright, so you kind of get this three-dimensional image. Now here, in this other structure, you basically have some type of animal cell. Alright. And here, in blue, alright, you have your nucleus. Alright. And in green, you basically have the actin, basically the skeleton of the cell. Alright, the framework of it. And then in red, you have all these other structures that are specifically stained that are the mitochondria. Alright, so for this cell, you can look at where the nucleus is, where all the mitochondria are in a cell, and you can look at the overall shape. And you can look at all these little cytoskeletal proteins of actin that make up the shape of the cell. And so this is a really cool process because you can actually add a chemical to a cell. And you can see how, if you're like looking at how it affects the mitochondria and how they're distributed, if you're wanting to look at how it affects the cytoskeletal structure of a cell, all right, you can stain it and compare, a cell with and a cell without that chemical and see how it changes the overall shape and structure of that cell, right? By tagging specific components. All right, so there's also a process called cellular fractionation, where you're basically taking a cell, you're splitting it open, and then you're separating out different cellular components. And so you can basically look at and separate these cellular components out and test them further. All right, so if you want to isolate ribosomes, or you want to isolate DNA, you want to isolate mitochondria, you can isolate those specific cellular structures and use them for further studies. All right, so here you have in a test tube cells. All right, and you can basically homogenize the cells. Basically, you're breaking them apart and basically chopping off all the internal bits and basically making up a big mixture. All right, so it's basically a cellular smoothie. All right. What you can then do is you can take this cellular smoothie. Now think about it. Think about a smoothie. All right, since I'm bringing this up. All right. Think about it. You mix it up in a blender, and let's say that you don't drink it right then. Let's say you put it in the fridge. And you want to let it cool down. Well, you come back, let's say an hour later, or you just set it up the night before. You come in the next morning, you open up the fridge. That entire sample of what you made in that smoothie, it's not homogeneous anymore, right? You actually see this phase separation of certain components. Things that are heavier tend to sink down to the bottom, and things that are lighter stay up top. Now, in this case, when you're talking about cell fractionation, what you use is a centrifuge. And what you're doing is you're spinning these samples down, and basically all the heavy cellular components are basically pushed down to the bottom. Now, depending on the rate of speed at which you're spinning your sample and the duration, all right, you're going to basically fraction out certain cellular components. All right, so for this instance here, all right, when you're spinning your sample down 100 times g, all right, so 100 times g is 100 times the force of gravity, all right, for 10 minutes. All right, your pellet, all right, basically the solid components down at the bottom, are going to be basically what we call cellular debris. All right, so cellular debris are what we normally call ghost. All right, and then you also have your nuclei. All right, so that would be in the first test tube. All right, in the second test tube, you spin it for 20,000 Gs for 20 minutes. And then you're left with mitochondria and chloroplasts at the bottom. All right, you spin it for 80,000 Gs for an hour. You get microsomes. All right, and then spinning it for 150,000 Gs for 3 hours, you get even smaller components. You get ribosomes. All right, so then you can take a sample of, like, let's say mitochondria, and you can basically test them with further experimentation. Now, later on we're going to do an activity in lab that involves this concept. Alright, but what determines the size of a cell? Right? Why aren't cells humongous and gigantic? All right. Why is it that something like, Godzilla, something the size of Godzilla is, biologically, an extreme? It's improbable. Well, it all has to do with the size of the cell. All right. Think about, why don't we have like gigantic bacteria? All right. When I'm talking about gigantic bacteria, there's like bacteria that are almost the same size as like eukaryotic cells. All right. When I'm talking about gigantic, I'm like, why can't you see bacteria with the naked eye? Why are they typically really, really small? All right. What's going to limit the size of a cell? Well, what do you think limits the size? What are certain factors that a cell has to contend with and deal with to remain alive? Well, for instance, you have something like nutrients. All right, nutrients have to get in, and waste products have to leave. All right, so nutrients have to come into the cell. All right, nutrients have to come in, and waste products have to leave. All right. Now, that's one of the major factors, is the ability to bring in nutrients, bring in certain things like gases, like O2 gas into the cell, and expelling out carbon dioxide. Alright, so what affects the ability of these components to enter and leave the cell? Alright, well, it has to do with, for instance, the cell surface area to volume. Alright, so notice that I said cell surface area, not cell area, but cell surface area. Alright, so what the cell is exposed to on the outside. Alright, what the outer surface is exposed to. Alright, so here, alright, to simulate a cell, alright, you'll do this in lab as well. Alright, but you have this cube. And this cube has a height, width, and depth of, let's say, 1 centimeter. Alright, so the total surface area of that cube is 6 centimeters cubed. Now, what about the volume? Well, the volume is basically, sorry, go back to that. Surface area, get ahead of myself, is going to be 6 centimeters squared, the surface area, and then volume is going to be 1 centimeter, alright, cubed for this instance. Alright, so notice that the surface area is centimeters squared and the volume is centimeters cubed. Alright, so if you take the cell surface to volume ratio, so you divide the total surface area by the total volume, you get 6. Alright, so notice that, okay, you basically cancel out most of your units. Alright, so what about a larger cube? Alright, so this time you have 5 centimeters. Alright, so this larger cube has a surface area of 150 centimeters squared. Alright, and you have a total volume of 125 centimeters cubed. Alright, so you're left with 1.2 as your surface to volume ratio. Now notice that you've increased the size of the cube, but that when you compare the surface area to volume of these two cubes, the smaller cube actually has a greater surface area to volume ratio than the larger one. Well, how is that possible? Why is that the case? Why does a larger cube have a smaller surface area to volume ratio? Well, here's the thing. Think about it. You're increasing the volume of the cell. Alright? You're increasing the volume of the cell. Alright, the surface area is also increasing. But which one's increasing more relative to the other? Okay? Your volume's increasing more relative to your surface area. Alright, so that's a limiting factor. Alright, so think about it. You have this little bitty cell, and then you have this really big cell. And think about it. Things have to get into the cell. Let's say the center of it. Alright? And so you gotta get things into the center of the cell, and you gotta get things back out. You gotta get things into the center, and things back out. Well, it's gonna take a longer time for things to diffuse into the center of the larger cell than the smaller one. It's gonna take a shorter time for things to diffuse out of the smaller cell than the larger one. Alright, so it has to do with how quickly materials can enter and leave that cell. Now, what about a cell that has the same total length, width, and depth as the 5 centimeter cube, but is actually made up of smaller 1 centimeter cubes? Well, in this case, your total surface area is 750 centimeters squared, your total volume is 125 centimeters cubed, and your surface area to volume ratio is 6. Alright, so think about, for instance, like intestinal cells. Alright, and when you're talking about like intestinal cells, alright, you typically see your little intestinal cells look like this. Alright, but when you basically zero in on one of these guys here, what you really see, you see these little microvilli. Alright, these little microvilli increase the surface area of absorption of that cell type. You have a greater surface area to volume ratio. There's more places for nutrients to be absorbed by. Alright, so in this case, this cell, even though it has the same overall length, width, and depth as the 5 centimeter cube, alright, it has a greater surface area to volume ratio because there's more surface area exposed to the outside environment. Alright, so let's think about, for instance, if I was to ask you to, in a classroom, alright, or, your bedroom, and I ask you to pick up an object from the hall and bring it into your bedroom, and to pick up, let's say, you put like 10 basketballs in your hall, and then you're supposed to bring them into your bedroom, versus if I ask you to pick up 10 basketballs in your hall and transport them individually to your neighbor's living room, or if you're in a classroom, all right, transporting the balls from one side of the room to another versus asking you to transport them from one side of the gymnasium to another. All right, you can tell there's a difference in the rate at which your ability to transport these things is dependent on the overall size of that cell. overall dependent on the size of that space. All right, it's going to take longer. Larger space, longer amount of time for things to get into the center and to move out from the center, back out into the environment. All right, so why exactly does all this stuff matter? All right, why do we care about, studying cellular structure? Well, up to this point, what we've talked about is biological molecules. So we talked about nucleotides, amino acids, and sugars, and lipids. All right, and that each of these little monomeric units make up things like DNA and proteins and cell wall components like cellulose. And then you have even larger cellular components like chromosomes and plasma membrane and cell wall. All right, so this is what we're going to start talking about now, is all this stuff. It's the individual components you find in a cell and the cell itself. But all this is tied back into biological macromolecules, their monomers, and the types of chemical bonds that hold these structures together, and the chemical groups that are contained in these structures, and the types of atoms that are contained in the structures. Alright, so everything goes back to basic chemistry. Alright, so you can't forget about basic chemistry, alright, because you're going to continue to come back to it over the course of this semester. Alright, so when you're talking about eukaryotic cells, right, your eukaryotic cells and your prokaryotic cells, alright, there are certain things that they have in common. Alright, and we're going to talk about those certain things. Now, we've already discussed prokaryotic eukaryotic cells briefly in the first chapter. Alright, but kind of come back to this idea. You talk about domains of life. You have bacteria, you have archaea, and you have eukarya. Alright, so your prokaryotic cells belong to domain bacteria, archaea, and then all of your eukaryotic cells belong to domain eukarya. So you have kingdom Plantae, kingdom Fungi, kingdom Animalia, kingdom Protista. Alright, so there's certain things that every single cell has in common. Whether it's prokaryotic or it's eukaryotic, there are certain features that you're always going to find present. Alright, so talking about these shared features. Alright, so prokaryotic and eukaryotic cells. Alright, well, what makes them a cell? Well, a cell has to have a plasma membrane, has to have a cell membrane. Alright, that makes up its overall boundary. It separates it from its external environment. So the membrane, you're separating the external environment from the internal environment of the cell, which could be completely different. Now, one of the things I want you guys to keep straight is the difference between the cytoplasm and the cytosol. Alright, so the cytoplasm is basically the area you find inside the cell membrane. Alright, so this is going to be all of your internal structures plus the semi-fluid-like material. Alright, something we call the cytosol. Alright, so cellular components plus the cytosol equals the cytoplasm. Alright, the cytosol is very similar to like Jell-O. Alright, the cytoplasm is like a soup. Alright, each of the noodles and the vegetables, all those things are internal structures. The broth of the soup is the cytosol. Both these things together make up the cytoplasm. Alright, now prokaryotic eukaryotic cells have ribosomes. Alright, or they have ribosomes and they also have chromosomes. Alright, so your chromosomes, alright, so if you're talking about prokaryotic eukaryotic cells, there are some differences. Alright, in terms of number of chromosomes. But in this case, in terms of chromosomes, they all have DNA as their source of genetic material. Alright, and they all have ribosomes. Ribosomes are basically little bitty protein factories. Alright, so if you're talking about the basic components that every cell has to have, it has to have a cell membrane. Alright, it has to have an internal composition. Alright, so you have a cytoplasm. Alright, you have chromosomes. You have to have some type of genetic material. And you have to have ribosomes. Alright, so these are some basic similarities. Alright, so what are the differences? Alright, well, you talk about a prokaryotic cell. Alright, they don't have a nucleus. Alright, so there's no membrane bound nucleus. Alright. What they have is called a nucleoid. Alright, so their DNA is just kind of sitting in the cytoplasm of the bacterial cell. Typically, it's centrally located, and it's going to be darkly stained. Alright, in this region that the DNA is found is referred to as the nucleoid. Alright, prokaryotic cells don't have membrane-bound organelles. Alright, they do not have organelles. Now, bacterial cells can have some very unique structures internally, alright, but they do not have membrane-bound organelles. Eukaryotic cells, they have a membrane-bound nucleus. Alright, so their nucleus has its own nuclear envelope. Alright, so basically you have the cell membrane, you have the cytoplasm, and then you have your nuclear envelope surrounding the nucleus of the cell. So it's its own little microenvironment. You have membrane-bound organelles. And then eukaryotic cells generally are much larger than your prokaryotic cells. Alright, so here you're looking at bacterial cells. Alright, so here you have an example of E. coli, and the E. coli cell has been broken apart, opened up, so that the genetic material kind of spills out. Alright, so here you're looking at the entire chromosome of E. coli. Now, your bacterial cells, they have typically one chromosome. Alright? When you're talking about eukaryotic cells have many chromosomes. So more than one. Alright, there are exceptions to the rules for bacteria, but they generally have one chromosome. Now, bacterial cells can also have what we call plasmids, which is basically extra chromosomal material. So extra chromosomal material. Alright, so your plasmids typically encode for things like antibiotic resistance or resistance to metal toxicity, or the ability to acquire alternative sources of nutrients, or even the ability to enhance the disease-causing ability of the bacterium itself. Alright, so we call this enhancing the virulence of the bacterial cell. Alright, so virulence factors. Now, bacterial cells have flagella. Alright, so they have flagella for motility. Now, the flagellum of a bacterial cell and the flagellum of a eukaryotic cell are different in terms of structure. Alright. but they are similar in how they function. All right. Bacterial cells have structures called fimbriae. These fimbriae are what they use for attachment. All right, so here you have E. coli cells of fimbriae. All right, the fimbriae of the E. coli cells are allowing it to attach to the surface of the epithelial cells of the intestine. All right, so for surface adherence, you have fimbriae. Bacterial cells also have things like capsules. All right, so these capsules are basically mainly protein and carbohydrates that are secreted outside the cell that make up this gelatinous glue, sticky substance that allows the bacterial cells to adhere to surfaces. Now, one important distinction is that bacterial cells have a cell wall. All right, and the cell wall of bacterial cells is made up of peptidoglycan. All right, so this here. All right, so just like in plants, the cell wall of plants is made up of what? Cellulose. In bacteria, the cell wall of bacteria is mainly made up of peptidoglycan. All right, so as the name implies, peptidoglycan, so proteins and sugars. All right, so basically you get layers of basically alternating sugar residues that get cross-linked and attached to one another by proteins, as well as glycosidic linkages. This cell wall functions as an external skeleton, all right, for the cell. It allows it to alter its shape and to expand. The cell wall of the bacterial cell also makes up and defines the overall, it aids in making up the overall shape of the bacterial cell. It's not the only component that makes up that affects the shape, but it's one of the major ones. All right, so here, looking at a bacterial cell, all right, so you're looking at darkly stained here, you have your nucleoid region, all right, where your DNA is located. You have outside, you have in the cytoplasm, you also have these little spherical dots of ribosomes. All right, you have your cell membrane, you have your cell wall, and then for this bacterial cell, you actually have, outside the cell wall, you have your capsule. All right, so here, you have your capsule. Now, looking at eukaryotic cells, all right, all these cells here, whether they're animal or plant or protist or fungi, they're all eukaryotic cells, all right? But notice how differently they look from one another. All right, so looking at an animal cell, all right, so here, looking at the features of an animal cell, all right, you want to familiarize yourself with this, all right, you want to be able to identify these structures and where they're located and what they do. All right, but here you can look at and see the cell membrane. You can see components like mitochondria and endoplasmic reticulum and Golgi bodies. You have your nucleus, where you have your nuclear envelope. You have your nucleolus. You have your chromatin. You can see all the cytoskeletal features, all the other cellular components. All right, and then looking here, we have a kind of a brief overview of the animal cell. Alright, so what about plant cells? Alright, so when you're comparing plant and animal cells, there are certain features that are found in animal cells but not plant cells. Alright, so plant cells don't have lysosomes. Alright, and plant cells don't have centrioles. Alright, now later on we'll talk about what these structures do. Alright, now your animal cells, alright, they don't have features that plant cells have. Alright, for instance, animal cells don't have a cell wall, animal cells don't have chloroplasts, animal cells don't have central vacuoles. Alright, so again, let's go back to this idea of a plant cell wall. Alright, so here you're shown an image of a cell, plant cell. Alright, you have your cell membrane, you have your cytoplasm in here, you have your chloroplast, and then you have this external structure makes up the cell wall of a plant cell. Now let's look at this a little bit more closely. Notice that the cell wall of a plant cell, alright, you have cellulose fibers, alright, many chains of cellulose fibers interconnected, and they're all kind of layered one on top of the other. And you have molecules of pectin, you have glycan linking chains, they're cross-linking structures, and these are attached to the cell membrane. Alright, so recall that cellulose, you have individual glucose monomers, beta glucose monomers, that are linked together through glycosidic linkages that make up one linear chain of cellulose molecules. And these cellulose molecules are interconnected by hydrogen bonding. They're held together in layers, and these make up your, basically, your plant cell wall. Alright, so you have your lignin, you have a hemicellulose, your cellulose. This makes up the cell wall of the plant cell together collectively. Now, looking at the cell membrane. Alright, so coming back to this idea of phospholipids. Alright, so recall that phospholipids contain your phosphate head group. Alright, you have your glycerol backbone, and then you have your fatty acid tail. Alright, so your phosphate is polar. Phosphate group is polar. And your fatty acid tails are made up of basically hydrocarbons, and they're nonpolar. So your phosphate head group faces the outside aqueous environment, and then your other layer of phospholipids face the inside of the cytosol. And then inside, in between, you have your fatty acid tails, right, because they're nonpolar, they're hydrophilic. Notice you also have proteins that are stuck in the membrane. Now sometimes you can have proteins that are stuck on just the surface, alright? But you can have some proteins that go throughout the entire membrane itself, that span across it. Right, transmembrane proteins. Right, things that we call integral proteins. Now we'll come back to these guys later on in the next chapter. Alright, but it's important to note that the plasma membrane doesn't exist purely of, or consists purely of just phospholipids. Alright, you have phospholipids, you have proteins, you have other types of lipids that make up this phospholipid bilayer. You notice that some of your proteins actually have sugar residues attached to them. Alright, so these are what we refer to as glycoproteins. Glyco implying sugar, and proteins. Now, looking at the nucleus. Alright, so the nucleus of a cell. Alright, so it's membrane bound. Alright, so it has its own nuclear membrane. Alright, in eukaryotic cells. Alright, so you have your own little nuclear envelope that surrounds the genetic material. Right now, inside the nucleus, right, you have your chromatin. Chromatin can either be densely packed, so it's darkly stained, or it's lightly stained, right, because it's loosely packed. All right, so what exactly is chromatin? Well, chromatin is the DNA, but it's a DNA that's actually wrapped around proteins that we call histones. And so the DNA, the chromatin fibers, wrap around, the DNA basically spoils around these histone proteins, kind of like old telephone cords, kind of twirl and wrap around. All right, your DNA kind of twirls around these histone proteins, basically forming something called a nucleosome. All right, and these nucleosomes can compact together. All right, they condense in structure, and this is what forms your chromatin fiber. Now, this chromatin in a cell can either be loose, all right, it's kind of spread throughout the nucleus of a cell. It's organized, but it takes up the entire contents in that nucleus, for the most part. And then right as a cell is beginning to go through this process of cellular division, your chromatin condenses into a chromosome, or in this case, chromosomes. All right. And then you also have this feature called a nucleolus inside the cell, inside the nucleus. All right, so looking at your nuclear envelope. All right, so here, your nuclear envelope has these nuclear pores, all right, openings. All right, so this is how materials enter the nucleus, and this is how materials leave the nucleus. Now, what exactly has to enter the nucleus? Right. Well, you have proteins, like, for instance, enzymes. They're involved in, like, DNA replication, they're involved in transcription, they're involved in editing a transcript, all right, editing messenger RNA, they're involved in repair of DNA, all right, so proteins that are involved in the regulation of genes that get turned on or get turned off. This is how your nucleotides enter the nucleus. All right. Now, what about what leaves the nucleus? All right. So, messenger RNA leaves the nucleus. But then you have ribosomes. Well, how do ribosomes fit in? Well, where do they come from? Here, you have the structure called the nucleolus. Alright, so the nucleolus, alright, is what's used to make our RNA. Alright, now you have proteins that also make up something we call ribosomal proteins. All right. Now, ribosomal proteins have to enter the nucleus and go to the nucleolus. And your nucleolus is responsible for synthesizing our RNA, which is a type of RNA called ribosomal RNA. Well, our RNA plus ribosomal proteins make up ribosomes. Alright. So the ribosomes are basically assembled and made in the nucleolus, and then they get transported outside into the cytoplasm through this nuclear pore. Now, your ribosomes. Your ribosomes, all right, you have a large subunit that has to be assembled, and you have a small subunit that has to be assembled. Now, this is one of those other differences, all right? So if you want to talk about prokaryotic, eukaryotic cells, prokaryotic cells, in terms of their ribosomal subunits, are much smaller than eukaryotic cells, all right? They function the same in that they're involved in making proteins that are just a little bit different. Okay. So ribosomes, all right, they're protein factories. That's what they do. All right. So how exactly are they involved in making proteins? Well, later on in the term, all right, we'll talk about the process of transcription and translation. All right. But here in this image, what you have is a messenger RNA transcript that's basically being fed through the ribosome. And as it's fed through the ribosome, you have other components that are involved in assembling the protein. All right, so the messenger RNA is moving through the ribosome. You have amino acids that are transported to the ribosome that are being basically assembled into the protein. All right, so basically, as this mRNA is fed through the ribosome, you have other components that are involved in making sure that, okay, that part of the messenger RNA transcript encodes for this amino acid. So that amino acid gets laid down, and then the next one, and then the next one, and then the next one, until you get a chain of amino acids that make up your protein. All right, now, when you're talking about ribosomes, you have two main types. You have free ribosomes, and you have bound ribosomes. All right, so your free ribosomes, all right, they're going to be involved in synthesizing proteins that are needed in the, they're basically needed in the cytosol. All right, now your bound ribosomes, all right, these bound ribosomes are attached to the endoplasmic reticulum. All right, so your bound ribosomes attached to the endoplasmic reticulum make up the, what we call the rough endoplasmic reticulum, or rough ER. All right, so proteins that are made by ribosomes that are attached to the endoplasmic reticulum are destined for other places in the cell. All right, so typically these are going to be proteins that have to be modified, like they have to have like a sugar attached to them, or they're going to be components of a lysosome, or they're going to be destined for the cell membrane itself. All right, so remember that the cell membrane has proteins in it, right? Well, how do these proteins get there? Well, they're made partially by the ribosomes that are attached to the endoplasmic reticulum. They're bound. And then you have proteins that are actually secreted outside the cell. All right, these are also made by the bound ribosomes attached to the ER, or the rough ER.